All right. Good afternoon. My name is André Brodkorb. I'm usually based at the Teagasc Food Research Centre in Moor Park, near Cork, in the Republic of Ireland. But like most of you, I'm working from home, so I'm right here in the living room of my house here in Middleton, which is not far from Cork. So if you will attend the Food Digestion conference in May of next year, some of you may visit Middleton because we have the Jameson distillery right outside our door. So today's topic is in silico food digestion, and we have three panelists, two speakers, three panelists altogether. I have Muireann Egan here, who is our communication specialist here in Teagasc in Moor Park, and Stephen Le-Feunteun, if you can show yourself, please. Stephen Le-Feunteun from INRAE, Choi-Hong from Greenwich, and Andrea Rivera del Rio from Wageningen. So without any further delay, I will hand over to Choi-Hong to lead today's webinar. And you are on mute. Very good.
Thank you, André. Thank you for the introduction. And today we have two talks. The first talk is given by Stephen Le-Feunteun, and he's from INRAE in France. And the title of his talk is Overview of the In Silico Modeling of Digestion. Stephen, please.
Yes, good afternoon to everyone. So just before my talk, I've prepared a few slides on our working group activities. So I will go through rather quickly. So I'll try to share my screen. Here it is. So is that okay? Do you see it?
Yeah, perfect.
So we are leading the working group, myself and Professor Choi-Hong. We have about 70 members, half with experience in modeling only. So we are not all modelers, and most of the represented expertise are related to food sciences and process engineering. Our main objective in our working group is to gather researchers interested in modeling food digestion and related mechanisms, to make the state of the art in mathematical modeling and numerical technologies for food digestion, and also try to initiate collaborative works. So for now, what we have been doing, so the first activity we have been running is to build a shared reference library. So this is done on Zotero online. And we also connect this reference library to what we call a gut map, which is open to all. So you won't find directly the full text of the paper, but you can manage to find the paper afterwards. And so the gut map is just a PDF file, as you may see on the left side of my screen, with URL links. So if you Google or go onto the INFOGEST website in the working group 6 slot, you will find the gut map, and you just have to click, for instance, on gastric emptying, and you will have a Zotero window, so by internet, that should pop up, and you can see in silico models related to gastric emptying. So this was our first activity. We also launched 3 review papers on complementary approaches. So the first one was related to the physiologically based modeling of food digestion. So models that have adopted a systems view approach, and more particularly on compartmental models. So here is a short small illustration of it. And I'm happy to say that it is now accepted in annual review of food science and technology. So the paper is in open access, so you can download it. So for now, it is only the uncorrected proof version, but the final version of the paper should come out in a few days or weeks from now. The second paper is on the modeling of enzymatic hydrolysis. So we are considering in that paper single nutrients and try to advance toward what can be done on complex foods in both static and dynamic in vitro conditions. So this paper is well advanced now and should be submitted soon. And the third paper is on the modeling of the biomechanics in the GI tract. So this time to deal more with food breakdown and mixing with the digestive fluids and absorption also, so all along the GI tract. So this paper was a bit in standby for a while, but we are now trying to give it a new start. And Professor Serafin Bakalis agreed to try to lead this task. So he would try to push forward for a new start for that review paper. And so to finish, in the future, what we want to do is to do practical works. So we wish to start building our own model, but we have plenty of questions before really jumping into that. So we are discussing so far on how to do what, etc. But this is under consideration. And another topic is in relation with Working Group 3, because we are discussing about how to build an in silico model of gut permeability, so at the gut wall and on nutrient absorption. So this task will be led by Choi-Hong from our side for Working Group 6. So it should start soon, hopefully. And so if you just want to join our working group, you can just email me and Choi-Hong. So do not hesitate. This is an open network, so everyone is welcome to join us.
Before you start, at the bottom of your screen you see a question and answer box. So if you have any questions, and I hope you do, type it in there. It would be great if you type it in before the talk ends. Okay, before the talk ends, and it's about 20 minutes from now. Okay.
Good afternoon, everyone. I'll try to do an overview of what can be done in the area of in silico modeling of digestion. So first of all, modeling should be viewed as a complementary tool to in vivo and in vitro experiments. So this can be used to evaluate the nutritional properties of foods and to draw hypotheses on the mechanism. So when we build an in silico model, we try to translate into mathematics these assumptions and use simulation to test our understanding of the phenomena, to make prediction, and also we should not forget it is a very useful way to gather the knowledge we have in a structured way. In the area of food digestion, in silico modeling is really a wide research area. There is a high diversity of methods, from statistical modeling up to computational fluid dynamics or mechanics. There is also a huge diversity of modeling objectives. People will be interested in enzymatic hydrolysis mechanisms, or food breakdown and mixing, or GI transit and nutrient absorption, and so on. And often these models are dedicated to one nutrient or food only. Others are using systems view approaches to consider all nutrients or peripheral organs, etc. So really, you should not forget that it is really a wide area and it is not so easy to do an overview of the field. So for this talk, I have chosen to show you a few examples on several topics that are covered by the area. So starting with the enzymatic hydrolysis and release of bioaccessible nutrients. Most common approaches are based on the Michaelis-Menten model and zero or first-order reactions with possible addition of a lag phase, of a maximum extent of hydrolysis, etc. Currently, most of the efforts are related to the integration of structural and compositional information to develop mechanistic relations between the model parameters and the food properties. So there are examples for all kinds of macronutrients. So for starch, for instance, models are trying to differentiate subclasses of starch with contrasted rates of hydrolysis to represent more or less resistant or more or less accessible fractions. For proteins, there have been lots of work on solubilized proteins, but probably now the effort concentrates on solid particles, how to consider their buffering capacity, how to simulate the diffusion of enzyme inside those particles, etc. For lipids, models are trying to account for the droplet size and the droplet size evolution. You will find models which consider a rate-limiting step for lipase diffusion in Pickering emulsions, for instance. And I will show you an example which tries to incorporate the fatty acid composition of the oil. I will now briefly highlight two examples that are both related to the intestinal in vitro digestion of oil emulsions. So the first example comes from the Catholic University of Leuven, in which they develop a multi-response model of lipolysis. So in vitro, they measured the release of free fatty acids, so the total of it at the top of the graph, but also the fraction that is solubilized into the bile salt micelles. They did the same for the monoacid glycerols and also monitor the increase in glycerol and the decrease in triacid glycerols. So they then proposed a reaction network made of three reactions. So one triacid glycerol can release the SN2 monoacid glycerols and two free fatty acids. And they also considered that the SN2 monoacid glycerol can be further isomerized into the position SN1 or SN3. And thereafter, this new monoacid glycerol can be further hydrolyzed into one glycerol and one free fatty acids. So they could validate their model on 28 independent datasets with very nice correlation between the predicted and experimental value. So clearly with that model, we can simulate all the main species that are met during in vitro lipolysis. I will go very briefly on that second example because it is highly similar to the previous one. What should be noticed is that they were interested in the bioaccessible fatty acids, so individual fatty acids this time. And the main result is that the in vitro fatty acid bioaccessibility kinetics reflects the substrate specificity of pancreatic lipase. So this is interesting for prediction purpose because this means that fair prediction should be able to be reached by knowing the substrate specificity of pancreatic lipase, the oil composition, and also the oil droplet size. With regards to the enzymatic hydrolysis mechanisms, we have seen that in vitro experiments and in vitro data are very useful to build in silico models. And now I'll briefly switch to another topic where I will show you that in silico can also be used to help in vitro findings to be translated into the in vivo situation. For this, I will show you two examples that are both related to the digestion of starch-containing foods. So the first example comes from John Monroe. As you may see, he observes the in vitro release of glycemic glucose equivalent for different loadings. And what is interesting is that he also estimated the glucose disposal rates from in vivo data. So just by subtracting the two curves, he was able to predict the glycemic response to various foods and also various loadings. And as you may see, there is a very nice correlation between the in vitro in silico approach and the in vivo data. So this example nicely shows that very simple calculation can be used to bring the in vivo results closer to the in vivo situation. So this second study is pretty much the same as the one just before. It is a study by Bellman and co-workers, except that this time they used a more elaborate in vitro procedure with an artificial mastication, a dynamic in vitro system this time with a dialysis membrane. So they measured the glucose and fructose concentration in the dialysate. And in the last step, they built an in silico model to scale back their in vitro observation into a more in vivo situation, in particular using a glucose-insulin homeostatic model. So they did that for 22 different foods, and as you may see, there is a very nice correlation between the predicted and measured human glycemic response for those foods. So this clearly shows that in silico strategy can compensate for some in vitro approximation. They can even extrapolate some in vitro findings into the in vivo situation. And so these examples are for starch, but probably we can expect that a similar procedure for lipids and proteins could be very useful in the future. We will now advance towards the modeling of the in vivo situation by highlighting what is being done actually with regard to the gastric processing and emptying. In first approximation, we can consider that gastric emptying follows an exponential decay, in which we can characterize a half gastric emptying time when 50% of the meal has been emptied from the stomach. If we look at the emptying rate in kilocalories per minute as a function of the caloric density of the meal, we clearly see that gastric emptying is also largely controlled by a nutrient sensing feedback. And this is not a new idea. Almost 50 years ago, Hunt and Stubbs already proposed a model to predict the half gastric emptying time as a function of the meal volume, V0 here, and the nutritive density of the meal. It is very recent that this idea was renewed in silico modeling of gastric emptying in a study of Moxon and co-workers in 2007, where they consider the gastric secretion, the food input. The stomach is viewed as a constantly stirred reactor. The small intestine is viewed as a plug flow reactor. And as you may see here, they are considering a nutrient sensing feedback to control gastric emptying. In their model, they also assume that gastric secretion is a function of the meal viscosity in the stomach. And using this model, they fit in vivo data on different glucose solutions of low to high viscosity and with also different loadings from 25 to more than 100 grams. So as you may see, their model was pretty good in predicting the gastric emptying kinetics for these different conditions. So this shows that in silico modeling based on those type of assumptions are pretty good to simulate the gastric emptying of liquid foods. So if the behavior of liquid foods can be fairly simulated, it is not the same for solid foods, which are characterized typically by a lag phase initially in the gastric emptying curves. So there have been some empirical models proposed in the literature from Elashoff, Siegel, and others, but we still miss a more mechanistic representation of this phenomena. They are under consideration or progress in different teams, but there is no real consensus on the way to do it, because you have to account for the sensing of nutrients in the duodenum, the liquid viscosity, the size, mechanical properties, and also the buffering capacities of solid particles. And most of the time you have a mixture between liquids and solids. So it's really uneasy to model this. So what I will show you in the next slide is more what is being done currently in slightly different techniques to try to progress towards a more mechanistic representation of those phenomena. First approach to better understand and predict the behavior of food in the stomach is to use computational fluid dynamics and mechanics. So pioneer three-dimensional models of the entire stomach dates from the early 2010s. So this model considered the case of a closed pylorus to investigate gastric mixing of liquid meals of different viscosity. More recently, there have been several papers from Harrison, who considered a pylorus that remained fully open, from Ishida, who investigated the coordination between the pyloric closure and the antral contraction, and how this affects the emptying of liquid meals. And more recently, a paper in which they consider gastric secretion and assume that gastric emptying rates are determined by the flux of calories delivered to the small intestine. So the same hypothesis as in Moxon or Hunt and Stubbs. And in that later paper, because they consider both secretion and reasonable gastric emptying rates, they could also try to predict the pH evolution within the stomach for different liquid meals, so water, orange juice, and whole milk. So we clearly see that in 10 years, or about 10 years, we were at a closed pylorus to investigate mixing, and now we can try to predict the pH within the stomach with gastric secretion and physiologically correct gastric emptying rates. Another approach to simulate the behavior of food at the gastric stage is to use chemical engineering approaches. So here is one example on the gastric digestion of meat by Jason Sikar, in which they try to consider a number of different phenomena. So they consider the diffusion of acid and pepsin into meat particles. In their model, the gastric fluid dynamic controls the penetration speed. They also include the meat buffering capacity, the pepsin activity as a function of pH to simulate the degradation kinetics. So with their models, they were able to estimate the relative influence of these factors and the uncertainties. And for instance, could show that the masticory capacity, hydrochloric acid secretion, and gastric motor are really key factors to understand the degradation kinetics of the meat particles. In contrast, the variation in pepsin content appears to have only little effects. So there are limits, of course, on these models. There is no mechanical particle breakdown, no biological feedback, but overall they try to consider the main mechanisms that should be included in a modeling of gastric digestion. And this model is probably well adaptable to other food products. Here is a second example on the gastric digestion of protein gel by Van der Smaan very recently. Just to show you that beyond acid and enzyme diffusion, we can also consider all the ions, so sodium, chloride, etc. And so this type of modeling is common to several groups. And here are examples of simulation reproducing trends observed in vitro with soy protein gel, where we can see the internal pH evolution, the volume of the gel particles that can swell or shrink, and also with the internal concentration in salt. So there is also a detailed discussion in that paper on how this type of modeling could be extended to model realistic in vitro gastric, gastric conditions. I will now briefly illustrate some systems view approach that are based on physiologically based modeling of transit and absorption. These approaches are generally based on compartmental modeling, in which the GI tract is represented by a series of reactors. So as you may see, we can consider the stomach, different fraction of the small intestine, the large intestine. Here is an example of observed versus simulated transit of a product within the GI tract with a very nice correlation. And with those models, we can also simulate the blood concentration of a product as a function of time. Here is an example where we use this modeling strategy to simulate the transport of dairy nitrogen from mouse to blood. So I will not enter the detail of the model. It is just to show you that by assuming contrasted gastric behavior of our dairy product, we could simulate fairly some concentration at the stomach exit, and we could also fairly simulate the amino acid postprandial concentration. Maybe one of the main results of that study is that dietary nitrogen is rapidly absorbed after being emptied from the stomach. And it also shows you that those modeling strategy can be very well adapted to simulate in vivo data. But if we want this modeling strategy to be useful for any type of meals, we clearly need to integrate some aspect of food composition. So this has been done in the animal science by Bastianelli first, and then revised and improved by Stratt. So here is a schematic view of the model developed by Stratt, where you will see dietary protein, lipids, sugar, starch, etc., and the different GI tract compartments. So clearly we can try to simulate the main product in the GI tract. So with their models, they found reasonably good prediction of the digestibility of the main food components, except lipids, and also the absorption profile of the studied nutrients. So this was glucose, amino acid, volatile, fatty acids. So this is just to show you that it is possible, in theory at least, to build a whole nutrient and almost a whole GI tract model using these modeling strategies. Although it is not published yet, we may highlight in here the model developed by George Van Aken, so one of our working group members. So he developed a compartmental model combining information from food, physiology research, and also in vitro digestion studies. So as input parameters, you will find the diet timing and some properties, such as the composition and some structural aspects of the meal. And the model will release a number of quantities that vary with time, so gastric pH, emptying, bile secretion, absorption of nutrients, of course. So you will be able to simulate the bioavailability of nutrients, and also it is linked to some feeling like hunger, fullness, satiety. So probably this is the most complete physiologically based models we know about. Here is one example of what can be done with that model. So in the graph, you see the results from the human study of Ederlink in 2012. So he studied the glycemic and insulinemic excursion following a bread and a pasta meals with the same content in carbohydrate, fat, and proteins. And as you may see, compared to the bread meal, the pasta meal showed a slightly reduced blood glucose excursion and a much lower insulinemic response. And here is results of the model. So with no fitting, so the model could nicely reproduce the trends observed in the in vivo data, just assuming that pasta has a longer oral and gastric retention time, and also because of lesser accessibility of starch in pasta compared to bread. So this is just an example, but we show you how powerful can be this type of modeling. I will now conclude my talk by listing some of the facing challenges. We have seen that many different things can be done in the modeling of food digestion, but clearly there is a need to better incorporate the food compositional and structural properties, and also the GI physiology, in particular the regulatory mechanism. We also face some methodological challenges. There are gaps of knowledge, so how to deal with them? What phenomena should or should not be taken into account? This is not an easy question because it partly depends on the modeling objectives. Also, how to combine different approaches that consider different lengths and timescales. So for instance, computational fluid dynamics and compartmental modeling are not that easily coupled. We also have a mathematical and computational issue for big models. And finally, the question of the experimental data to compare and validate in silico models is also a real bottleneck when building and trying to validate a model. To conclude, in silico modeling is a rapidly growing research area with a high diversity of objectives and approaches. Overall, it is highly complementary to in vitro and in vivo studies, in particular to gather and structure the available knowledge, to test our understanding of the mechanisms, and also sometimes to extract hidden information from experimental data, in particular in vivo data, and of course to make predictions. So there have been important success or promising results in several areas related to food digestion, but important challenges remain to be overcome before whole body and whole nutrient in silico approaches could predict the effect of food on physiologic and metabolic responses. To finish, I would like to thank all the members from our working group because this talk was largely inspired by the activity we have been running and the discussion we have in our working group, and also for sharing their slides, Tara Growet and Sarah Verkamping, George Van Aken, Jejan Sikar, and Alain Condoyan. And I thank you all for listening.
Thank you very much, Stephen.
So one question is, to what extent does the lipid bioaccessibility correlate to in vivo lipid bioavailability? I'm not that sure, but we have been working on this with a PhD student a few years back. And so although it is not published, what we observed is that the lipid bioavailability was very well correlated to the lipid bioaccessibility. But you need to account for the fatty acids that are already circulating within the blood plasma. So let's say you have a base lane before eating, and so during the postprandial periods, some of the fatty acids will decrease from your blood, while others are coming from the digestion system. So I would say, yeah, more work is still needed, but probably it is okay. There is also a question on by Anne Meunier. Any explanation to the poor prediction of lipids digestibility in the model of Strat? So this model was for pigs. I don't really remember, but this paper is really detailed. So there are plenty of very interesting things in that. So I'm pretty sure they are discussing this. Probably in my memory, I'm not sure they are accounting for the lymph. So I'm not sure whether the fatty acids, the way the fatty acid is correct. So if you see other, Choi-Hong, do not hesitate.
I think we have time to go for one more answer question.
Okay.
And I think then afterwards we should switch to the other speaker.
Yep, go on. One more question, Choi-Hong.
So there is one more question. So this is from a PhD student. So he would like to know which wall and encapsulation technique are most suitable for target delivery of flavonoids to the small intestine.
So for this, I'm not so sure. Some people are really expert in that field to encapsulate using oil some lipophilic compounds. So I'm not sure to what is the correct answer. This is really an area of research actually, but I'm not sure I can answer this question. Sorry.
Just one remark here. First of all, thanks for the question. And we have somebody from Brazil here, which is great, live attendant. We will answer all questions afterwards. So if you don't have a chance to answer the question now, we will answer afterwards. And that's probably one of those questions that will be answered in writing. Okay.
So I will now pass the chair to Stephen, and who will chair the second talk.
Yeah, great, thank you, Choi-Hong. So the second talk will be done now by Andrea Rivera del Rio. So Andrea is a PhD student at the moment at the University of Wageningen in the Netherlands. And so she will talk about the modeling of protein digestion in liquid and solid meals.
Hello, my name is Andrea Rivera del Rio, and today I have the pleasure of presenting to you our approach to modeling protein digestion of solid and liquid meals. Before I start, I would like to thank the organizers of this webinar for giving us the opportunity to present our work to you. By the end of this presentation, I hope I will have been able to show to you that using dynamic mass balances is a simple and intuitive approach towards modeling food digestion. And since in this presentation we are focusing on the differences between solid and liquid meals, I want to show to you how, by incorporating antral grinding to our food digestion model, we can describe relatively well the observations that have been done in vivo when comparing meals of identical composition but different physical states. You can expect that the digestive fate of a so-called solid meal, such as this fruit salad, will be different when it is consumed as a smoothie. So the composition of these two meals is identical, there's just a difference in the structure and the particle size. This is nothing new. In fact, it has been researched extensively in vivo studies. And the reason for the different digestive fate is a concept called gastric sieving. When we consume a solid and liquid meal, so a meal composed of a solid and liquid component, it arrives as such in the stomach. The liquid is emptied first and faster into the small intestine. The solid, however, needs to go through a different process. It first needs to stimulate the antrum to start contracting after a lag phase, and once the contractions have started, so does the grinding. Once the particles have been reduced to a size small enough to go through the pylorus, gastric emptying of the solid component can start. On the other hand, for the homogenized monophasic meal, what we have is that the blended meal enters as such in the stomach. Gastric emptying starts, but the difference here is that the chyme that enters the duodenum is actually having a higher nutrient density. Therefore, gastric emptying is delayed. The residence time of the total meal is longer in the stomach, and thus satiety is increased. We have incorporated these concepts into a model created for educational purposes at the university. This model is designed to simulate protein digestion. Here, what we do is simulate intake within the stomach, breakdown of the particles, as we have mentioned before, gastric emptying into the duodenum. Also, what we do is simulate secretions from the parietal cells and chief cells into the stomach, as well as from the pancreas into the duodenum. Within the different compartments, hydrolysis of protein and peptides, as well as transit through the small intestine and absorption and digestion of nutrients. This is the flow diagram of our model. It includes the processes that we deem most relevant to protein digestion. And this is where the mass balances come in. Each box in this diagram represents a pool of a nutrient or a secretion, and each arrow represents a flux, again as transit, secretion, or transformation of a nutrient. We assume that the contents of each compartment are ideally mixed. For this model, we have used SMART. This is a software that was created at Wageningen University. And this is where we solve our mass balances by using ordinary differential equations. This is a software so simple to use that students can pick it up how to work with it just within one afternoon during the food digestion courses. The model that I will show to you is greatly inspired by a study from Marciani and others in 2012. In this study, the participants consume one or two meals, both composed of chicken, vegetables, and water, either as a solid and liquid meal or a soup meal. For the purposes of simplicity in our model, we have translated this into a pool of protein, a pool of non-protein, and a pool of water. And this is how it is included in our model. We have described the rate of gastric emptying as a function of energy, as you can see in this function, which includes the caloric density of the meal. For the solid liquid meal, however, water is emptied as a non-caloric component. And as we have mentioned, the solid components first need to go through a lag phase before antral grinding can start. Antral grinding is described as a zero-ordered function in this case. And then gastric emptying, once the particle size has been reduced to a size small enough to go through the pylorus, gastric emptying can start. And this is also described as a zero-order function of 2 kilocalories per minute. In the Marciani study, they quantified gastric volume by studying scans from an MRI. And what they showed was that the soup, for example, was emptied at a constant rate from the stomach by looking at the gastric volume. On the other side, for the solid liquid meal, there is initially a sharp drop in the gastric volume, followed by a slowdown drop in the gastric volume. This is interpreted by interpreting the scans from the MRI as a fast emptying of water, where solids remain in the stomach, and then are slowly emptied into the small intestine. For us, this is the result from our simulation. We observe as well the constant decrease in gastric volume or constant gastric emptying, and we also observe the biphasic nature of the solid liquid meal, with an initial sharp drop followed by a more slower gastric emptying by the solids. However, if you look at this graph, you can see that the gastric emptying of the liquid component lasts for longer than compared to the in vivo study. We attribute this to the presence of gastric secretions. The gastric acid in this case is composed of water, acid, and pepsin. These are secreted into the stomach with the arrival of the meal. We know that up to 2 parts of gastric juice are secreted for 1 part of food. So this is a quite significant secretion compared to the meal size. And this is how we can explain that longer, fast decrease in gastric volume, but because of the presence of a significant amount of water that takes long to empty. Now that we're talking about secretions, let's talk about intragastric pH. We have defined intragastric pH we have calculated it using the concentration of free acid in the stomach. The inputs of acid in the stomach come from the secretion from the parietal cells, as well as from the pH of the food. Since we're talking about a chicken soup, of course the pH cannot be lower than 5.5 or so. Therefore, this is not a significant contribution to the acid pool in the stomach. As protein is consumed with the meal, protein is one of the components with the most significant buffering capacity in the meal. And this is also described in our model. So we have an outflow from the acid pool into the protein to account for the buffering capacity. The protonatable amino acids are considered as the conjugated base to a weak acid, which is the buffered protein. As pepsin-catalyzed hydrolysis starts, the cleavage after each cleavage of a peptide bond, the protonation of the amino group is also considered. This is the second outflow from the acid pool. Lastly, the gastric emptying also contributes to the reduction of the acid pool in the stomach. Now, by solving this mass balance, we calculate the pH. And this is what we obtain. This is somewhat what we can find for intragastric pH after consuming a meal, a sharp increase followed by a decrease. Of course, this decrease doesn't look exactly as expected. You can see these discontinuities are a bit strange to describe acid pH. The reason for these discontinuities is the different events that we have mentioned as the outflows from the acid pool. And we all know that intragastric pH is a very, very complex phenomenon. And not only is it changing over time, but also spatially within the stomach. So maybe this is an indication for us to see that describing the pH in a single compartment for the stomach is probably not the best approach. Nevertheless, this was also studied in 79, and a study which also compared a homogenized meal to a solid liquid meal. The one on top is the homogenized meal. And they show that the pH of the homogenized meal remains always slightly higher than the solid meal. This is also what we observe from our simulation. Moving on to pepsin-catalyzed hydrolysis, we have defined it as a function of pH and the concentration of pepsin. With the secretion of gastric juice also comes pepsin. Thus, the pepsin amount, the size of the pool in the stomach increases over time. If we translate this to concentration, we can see this sharp increase, and this is explained by this increasing pepsin amount in the stomach, but also by the reduction of gastric volume that we have discussed before. With this function, we can calculate the proportion of active enzyme from the total enzyme present in the stomach. As we know that pepsin activity depends on pH. So we can calculate how much of the total amount of pepsin is active at a different time points within the residence time in the stomach. And then we can calculate how much the hydrolysis rate of protein in the stomach as a function of the pH, and the concentration of pepsin. And we can see how the hydrolysis of protein increases over time. We can use this same concept to talk about residual pepsin activity in the duodenum, because you can expect that when acid chyme leaves the stomach and enters the small intestine, as well as pepsin, there is a moment where there is some activity also of pepsin also in the small intestine and not only in the stomach. Now that we're talking about the small intestine, let's talk about the hydrolysis taking place there by the endopeptidases such as trypsin, chymotrypsin, elastase, etc. When water arrives in the duodenum, for example, with the solid liquid meal, where by gastric sieving water appears first in the small intestine, there is no stimulation of the nutrient-sensitive enteroendocrine cells. However, when nutrients arrive in the duodenum, the pancreas is stimulated, and then the pancreatic juice secretion can start. And with it, we can have the secretion of endopeptidases and bicarbonate. As the digestion continues, and so those transit through the small intestine, endopeptidases as well as nutrients continue to flow through the small intestine. So what you can imagine is that when the blended meal enters the duodenum, almost as soon as it is entering the stomach, the pancreatic juice secretion starts and also does the transit into the different compartments of the small intestine. For the solid liquid meal, because of the delay in gastric emptying, you can expect that endopeptidases will not be secreted until after antral grinding has started. And we can also describe hydrolysis as a function of endopeptidase concentration because of this dynamic pattern of secretion. So with this model, we can know at different time points how much protein is present or how much peptides are being formed or as products of hydrolysis of protein in different compartments at different time points. We can also know something about amino acid absorption. And as you can expect, because of the later arrival of nutrients into the small intestine, that also amino acid absorption happens with a delay compared to the blended, in the solid liquid meal compared to the blended meal, as you can see here. Modeling food digestion is a work in progress. We have used this model that I have shown you today as a template, for example, to study what happens when we consume consecutive meals. In regards of protein digestion, for example, when a meal contains slow and fast proteins. Or for example, if we have a meal of plant origin, whether we have trypsin inhibitors or not, and how much they can affect small intestine digestion. We have also created a model for carbohydrate digestion, and specifically about the effect of a non-competitive inhibitor of intestinal sucrase, and how much this can affect, reduce, or delay glucose absorption into the bloodstream. But perhaps an interesting approach to use this in silico models can be to make our work in the lab more efficient. Maybe we have gathered quite some data on an in vitro study, and we want to know what would happen if we changed the condition. We can incorporate this into our model and make our work in the lab, our in vitro studies more targeted and more efficient. This brings me to the end of the presentation. I have shown you how by using dynamic mass balances, we can have a simple and intuitive approach towards modeling food digestion. And this can be an entry point for someone who doesn't have so much experience with modeling, as do students. We have seen how by incorporating antral grinding to a protein digestion model, we can describe relatively well the observations that have been done in vivo when comparing meals of different physical states. I would like to thank you all for your attention, and I get once more to the organizers of this webinar for giving us the opportunity to talk to you today, and to everyone who has contributed to this work. Thank you so much.
So thank you very much, Andrea, for this very clear and nice talk. So we have time for a few questions. So I'm going back to the question box. First question is, why does water, a non-caloric food, takes longer to empty, especially if gastric emptying is based on 2 kilocalories per minute? But maybe I'm not so sure whether it was with your blended.
I think it was with the solid liquid meal that we see this biphasic behavior of in the curve. And well, what we think is that since we are incorporating also gastric fluid secretions, we are doing this also in a zero order with a zero order function. And what happens is that perhaps there's just too much contribution of that, or that the rate that we are using for water emptying secretion is maybe slower than it should be. So we think that this is why this is taking longer than compared to the in vivo study.
Okay. Another question is about gastric pH. Could you please comment on what is meant by intragastric pH, since it can be quite inhomogeneous? So how do you deal with that?
Yes, that's absolutely correct. Well, for us, the way it's calculated in the model is the concentration of free acid, or just the acid that hasn't been going either into the small intestine to account for the buffering capacity or for the protonation after cleavage of a peptide bond. That amount as a concentration, so essentially divided by the gastric volume. So of course, this is a very, very far-fetched approach, because it's not only an inhomogeneous digesta, but it's also, as we say, changing not only during time, but like across the time, but also spatially. So yes, this is absolutely correct. That's why I think that it's necessary to incorporate both the spatial aspect, but also the inhomogeneity of the meal or the digesta.
Okay, great, thank you. Do we have time for last one, André? Yeah. So this one is from myself, but you were talking at the start that you were considering particle size possibly greater than 2 millimeters. You also showed us how do you deal with the pepsin hydrolysis activity, let's say, as a function of pH, etc. So in your model, is there a relation between the hydrolysis you calculate and the evolution of the particle size? And if yes, how do you do that?
Well, in the figures that we showed today, not. But this is something that we, it's sort of a loop that we are trying to incorporate, because essentially at the moment, protein can be hydrolyzed from both protein pools, the big particles and the small particles. And these now becomes part of the peptide pool. So what some of the things that we have tried to incorporate is texture of the meal, and how the diffusion of the enzyme can be taken into account, so that the protein is hydrolyzed from outside in, and then this also contributes to the particle size reduction.
Okay.
Yes.
Great, thank you very much. I think I have asked all questions in the boxes, so
Excellent.
So I'll leave you the floor, André.
Excellent. So first of all, thank you both for the very nice talk. Just one last remark from my experience with working with you. The guys in the working group 6, they're always looking for in vivo data, and they're looking for the raw data. So if you are working in vivo digestion, and if you publish it, publish as much as you can in the supplementary material. Okay, the raw data are crucial. They're really needed for any simulation. Okay, so I just have 5 more slides. Some announcements for the next events. So the next event will be on the 15th of March, so not the first Wednesday in March, but the 15th of March. As a guest speaker, and Caroline Offila will moderate, lead this special webinar on starch digestion, or the working group starch digestion, in particular about the amylopectin and starch functionality. The guest speaker is Professor Serve Perez from France. Subsequently, the next webinar, the next regular webinar will be on the 7th of April, so the first Wednesday again, in April, and the topic is in vitro digestion methods. And last but not least, we have decided to, in place of this year's Food Digestion Conference in Cork, which will happen in 2022, so 1 year later, we will organize a virtual food digestion conference on the 6th and 7th of May. So that's a Thursday, 6th of May, probably the afternoon, and then the next morning, the 7th of May in the morning. So details will be announced soon. Just a reminder, we are dealing with 6 working groups, and the themes of the conference will be these 6 themes, and the speakers, the working group leaders have the responsibility of choosing the speakers. So we will have 6 sessions and 1 more, and that will be on the Friday morning for our colleagues and friends in Australia and New Zealand, so that they can join us live. If you want to be a part of the INFOGEST network, please send an email to Nathalie Lamar, and I see that you see that on the next slide, that you want to join the INFOGEST network. Again, it's an open network, and indicate which working group you want to be part of, and you can be a part of multiple working groups. And Didier Dupont, the chair of the INFOGEST network, sent an email out this morning with I think a doodle pool. So please open up the doodle pool and indicate your preference, your allegiance with a working group, and you can be in multiple working groups, so that the communication will be a bit more targeted, because there are a number of ring trials ongoing and planned for this year. So please answer the email by Didier Dupont. This is the email Nathalie Lamar. Oops, I'm sorry. Nathalie Lamar sent her an email to join and indicate the working group. Just a reminder here, we have a LinkedIn INFOGEST group. Just look for INFOGEST and LinkedIn, and if you want to tag a paper, or if you're looking for a job advertisement, just put @INFOGEST, so @INFOGEST at the end, and anybody that subscribes at the moment, or more than 500 people, will get this notification. Okay, and again, a reminder that all webinars are published on YouTube. So if you go to YouTube and look for in vitro digestion and INFOGEST, you will eventually find this. So that's my last slide. Thank you for tuning in again, and see you again on the 15th of March, slightly different time, so 11 o'clock a.m. I'm sorry, that's a mistake, a.m. Irish time. Until then, goodbye and slán.
Goodbye, and thank you, André.